And since then, he's quite well known for his work on Australian marsupial genomics. In particular, he had a science paper a couple of years ago with Liz Murchison where they outlined the cell of origin for the Tassie devil facial tumour disease. So you may have seen Tony on TV at that time. So he's worked a lot on next-generation sequencing. He currently holds a joint appointment with the Eu-myc and holds the Gully Fellowship there. And his work now very much focuses on cancer genomics, and the talk today will be about that. And today he'll be covering two topics in his talks that are aspects of cancer genomics: modelling of catastrophic chromosomal rearrangements in some cancers, and this is work that's currently under second round review with a fairly prestigious journal. And he'll also talk about some software that was recently published in Bioinformatics called Socrates to also detect chromosomal breakpoints. So welcome, Tony.
Thanks, Melanie. So this beautiful fish image here I'd like to talk about for a minute. This is an image of chromosomes from a Liposarcoma cell line. And if you haven't noticed already, there is a giant chromosome sitting there. And these things are called Neochromosomes. And one of the things I'd like to talk to you about today is the process that we undertook in order to discover what the mechanisms of forming and the evolution of these neochromosomes is. So I thought I'd start by saying a few things more broadly about my lab. The research focus in my lab is really bioinformatics, and particularly analysis of protein and nucleotide sequence data. So we develop, I guess a major part of what we do is we develop novel algorithms or bioinformatics methods as they're required to make sense of data. So this is always motivated by data that emerges from collaborations. And we're pretty diverse. We've applied our methods and worked on problems in malaria and also marsupial genomics, as Melanie mentioned. But these days we're primarily focused on cancer. So I'm going to talk about two projects today that Melanie's already outlined. The first really describes the formation and evolution of the giant chromosome that you saw in that first slide. The second is going to be about mapping transgenic insertions in mice. The interest here is really still evolution, but the first step in understanding evolution in transgenic mice is really mapping the transgene. And it's surprisingly not straightforward to do this. Along the way, what I want to do is talk about some of the methods we've developed to make sense of the data that we deal with. So in particular, I'm going to talk about some boutique methods that we've developed to analyse DNA copy number from next-gen sequence data. Two different methods of analysing genomic fusions in sequencing data, and also a kind of an approach to inferring the life history of tumours from individual sequence samples. So the first project is, as I've said, is about neochromosomes, these giant chromosomes that appear in some cancers. And this image over here shows two neochromosomes. One is a giant linear neochromosome here, and the other is a ring. So neochromosomes can take these two forms. The linear chromosomes are like normal chromosomes, except they're massive. They have centromeres and telomeres. The telomere only in the linear form. The ring chromosomes have a centromere but no telomere. And these are examples of chromosome scale mutations. And chromosome scale mutations have been a focus of recent interest. And they include mutations like double minutes, a hypermutation feature of some tumours called contagious, and also large-scale chromosomal shattering, which has been called Chromothripsis. This is yet another example of a chromosome scale mutation that is little studied. So neochromosomes are found in about 3% of tumours, but they're very common in certain kinds of sarcoma. So in this talk, I'll focus on well-differentiated liposarcomas, and about 85% of liposarcomas have neochromosomes in them. But they also appear in some hematological malignancies. What we know about neochromosomes up till now has really come from low-resolution studies. So from studies like this, which is a multicolour fish image from a well-differentiated liposarcoma cell line, we've learnt that neochromosomes are composed of material from multiple chromosomes. These are the neochromosomes over here, and they look like coloured socks. And you can see that they contain chromosomal material from many different chromosomes. In well-differentiated liposarcoma, chromosome 12 is always present and always very highly amplified. And it's known also that these neochromosomes harbour highly amplified copies of known oncogenes, including MDM2, CDK4, and HMGA2. And it's thought that most of the key oncogenic events that occur in these tumours lie on the neochromosome. But it's not understood how the cell tolerates the formation of these things. So on the basis of these low-resolution studies, it's been proposed that neochromosomes form through a process of breakage-fusion-bridge. Now, there are two flavours of breakage-fusion-bridge that I'll explain to you briefly. The first is linear breakage-fusion-bridge, and this was initially identified by Barbara McClintock in maize in 1941, so in the thing that you make beer from. Linear breakage-fusion-bridge starts with the loss of one telomere, and then during replication, a second identical chromosome is formed, and because nature abhors naked DNA, a fusion occurs, forming a chromosome with two centromeres. Now, at anaphase, the mitotic spindles grab the centromeres and rip them apart, and a double-strand DNA break has to occur between the two centromeres. This leads to the daughter cells inheriting different chromosomes, missing telomeres, and the cycle continues. So in doing this, breakage-fusion-bridge can amplify material or delete material on these chromosomes. The second flavour of breakage-fusion-bridge is called circular breakage-fusion-bridge and operates when both telomeres are lost. So you start with loss of two telomeres or telomere association, and a ring is formed. The ring replicates, and then something quite special can happen. During mitosis, crossovers can occur between these two rings. If an even number of crossovers occur, the mitotic spindles pull the rings apart, and they separate quite cleanly. But if an odd number of crossovers occur, you end up with a dicentric ring once again. At anaphase, these have to break, and you end up with different rings being inherited by the daughter cells. And this process allows duplication and deletion of material as well. And it's a cycle. So as long as you get odd numbers of crossovers, you will get amplification and deletion. So in order to characterise neochromosomes at higher resolution that's been done to date, and to investigate how they evolved, we flow enriched a sample of neochromosomes and then sequenced them. So we started with 5 cell lines, 2 of them related. So the cell line 449 comes from a primary tumour of a liposarcoma, and 778 is derived from a recurrence that occurred 12 months later. The other 3 cell lines that we looked at were independent. And so this just shows how the fish works. So these are the how the flow sorting works. These are the normal chromosomes down here, and 778 contains 2 neochromosomes, 2 isoforms that are quite similar to each other, slightly different in size, and both of these were captured and pulled together. The same was done for the other cell lines. We then sequenced this enriched neochromosomal material to between 5 and 30 times coverage. So our coverage differed between the different samples. We also performed RNA-seq on the cell lines and additionally sequenced 2 patient primary tumours without enrichment to about 30 times coverage. And in addition to the sequencing, Owen Marshall, who was then at MCRI, undertook a number of FISH studies on the cell lines, as well as ChIP-seq. And I particularly wanted to highlight this so I could say fish and chips in my talk. Notably, this data was really built up over a period of several years. So where initially we were sequencing these cell lines to quite shallow depth, as time progressed, we were able to easily generate more and more sequencing data from this. So our initial focus was on single nucleotide variants and indels. And so we start by aligning all these reads generated from the neochromosomes to a reference genome and look for SNVs and indels using standard tools to do this. And what we find is there are no recurrent single nucleotide variants on the neochromosome. There are a few things that looked interesting that were predicted as damaging, but following these up, there was really nothing interesting, at least on the neochromosome. We didn't really have power to look at the other normal chromosomes in this study. There were several fusion genes, and I guess the most interesting thing about these is that they contained novel exons. So exons that are not parts of normal genes, but are encoded in introns. But knocking these down, we see no effect on the tumour cell lines, and so the conclusion is they're not functionally important. And none of them are recurrent. So since the low-resolution studies really highlighted the importance of amplified oncogenes in neochromosomes, our main focus turned to copy number analysis. And this shows a plot of what we call read depth. So every dot here in this plot is the number of reads counted in a 5 kb window for a set of windows that span the genome. And there's a few notable things about this. Firstly, you'll see that this is chromosome 12. The whole chromosome is actually sequenced. The flow sorting doesn't isolate the neochromosome, it just enriches it. And so we end up sequencing at low coverage the entire genome. This amplified and enriched stuff here is the neochromosome. It's part of the neochromosome. The other thing that you'll notice is that it appears that there are waves in the data, and this is a bias due to GC enrichment, or the effect of GC bias in sequencing. And this is quite correctable. So what we do is, if we plot the GC content of these windows, and again the numbers of reads in each of these windows, we get a plot that looks like this. This would be familiar to bioinformaticians in the audience, probably. This central fat band is a result of all this background material. And then these bands that sit on top, they correspond to different copy numbers in the tumour. And what we want to do is try and fit a curve, a low S curve, through the centre of this cloud, and we use that to correct the GC bias in the data. So that's the first part of the sort of the correction process that we go through. Whenever we want to deal with copy number accurately, we need to subtract out this background, and that's reasonably straightforward to do. I actually like to leave it in because it's quite informative, and there are clearly things going on in the background. So all of the plots that you'll see will retain this background, but where it's important, particularly for calculation of neochromosome sizes, we subtract this out. The final step is the tricky bit. How do we calibrate copy number in data where there's enrichment by flow sorting, as well as all this mess in the background? And the way we do that is to turn back to the low-resolution studies. So by looking at FISH images like this, we're able to tell that there are regions of low copy at the ends of the neochromosomes, so corresponding to chromosome 22 and 7, that provide telomeric caps on the neochromosome, and we use this information to provide a calibration. And so here you see the copy number plot for chromosome 22, which sits at 1 copy and lies at the telomere. So this allows us to convert kind of this mess into copy number. And you see that our background material sits well below 1 copy, which makes sense. And we have this very highly amplified material that forms part of the core of the neochromosome. These are 2 plots of chromosome 12 from 2 different samples. The first is the 778 cell line, which is the thing I'm going to show you the most pictures of today. But everything that we notice or discover about 778 held up in the other cell lines that we examined. So all of the neochromosomes show this similar striking pattern. So on chromosome 12, you see this focal amplification of material. There are hundreds of amplified genomic regions present in these plots. In 778, there were over 300 coming from most of the chromosomes of the genome. And on chromosome 12, there are about 50. If we zoom in on these focally amplified genomic regions, we see that they're highly structured. So they're internally rearranged and with different copy numbers even within the amplified regions. And the question emerges, how are these things connected to each other in the neochromosome? So to determine that, what we need to do is look at the genomic fusions that are present, how these blocks are connected together, or what are the rearrangements that the neochromosome undergoes. To do that, there are a number of ways you can get at this information. A very simple approach is to look at paired end sequencing data. So this shows reads coming from the tumour genome, and these are reads that span a breakpoint or a fusion. So the blue reads sit on one side of the fusion, the red reads sit on the other side of the fusion. If we sequence this bit of DNA and then map the reads back to the genome, they may align to different chromosomes. So the blue chromosome and the red chromosome. Alternatively, this might be an intrachromosomal rearrangement and they simply align too far apart from each other to make sense. So we're looking for patterns like this in the data. This is not the only kind of evidence that you can look at, but it's a simple, direct approach, and it's where we started. So this is now a view directly of the data. We've got at the top the copy number profile, and we're really looking at the edge of an amplified region here. These are the discordantly aligned read pairs that we've looked at to try and figure out how the genome is rearranged. And there's some really striking things about this. So these spikes here are highlighting the presence of a fusion. But you can also see that there's a lot of different colours in here, and that represents noise. Most of the reads here are singletons. They're just a read that comes from 2 different chromosomes that you see only once. And the most likely explanation of these reads is that they are formed in the process of sequencing. They're chimeric. And at the bottom, what we've done is we've applied, I should also point out, up here is what we call a pile-up of these discordant reads. So essentially what we're doing, it is every nucleotide, we're counting how many reads provide evidence for a fusion. Fairly obviously, where there are spikes, there are fusions. And this is really just background noise in between. So at the bottom, what we've done is we've taken Breakdancer, which is a publicly available fusion detection tool, and I'd characterise it as an oldie but a goodie. So it was published in 2009. It's known to have a high false positive rate, but it generally is supposed to be pretty good. And these are the calls from Breakdancer, these little blue lines. And they don't match up very well from where you would expect the fusions to occur. And in particular, the fusion right at the edge of this amplified region, which naively we thought might be important, and it turns out that it is pretty important, was systematically missed by Breakdancer. So Breakdancer applies a number of filtering steps that for some reason systematically removed these fusions that sit right at the edge of these amplified regions. So clearly we had to do a little bit more than use an off-the-shelf tool in this analysis. So we developed what's really a boutique fusion caller. This is not something that you would apply to tumour data generically, but was really customised for the problem here. So we started by predicting amplified regions from the copy number data, essentially segmenting out these regions. We then identified clusters of discordantly aligned reads. So these are our discordantly aligned reads. If you plotted the read locations in chromosome A versus chromosome B, it might look something like this. So these chimeric reads that are just a source of noise would appear as isolated singletons, and a real fusion should appear as a little cluster. And so that's what we try and detect. But we do this extra step of if we detect clusters that are sitting outside of the amplified regions, we discard them. So we're only focused on the neochromosome. And that turned out to work quite well. And now we're finding all of these edge fusions, these fusions that lie at the edges of our amplified regions, we're able to detect. And we got about a 90% true positive rate when we performed validation tests on a subset of the data. So when you put all that together, the copy number information and the fusions, you get a picture like this. This is for the 778 neochromosome. It has over 500 genomic fusions on one chromosome. We showed this to the folks at the Sanger Institute and they said they'd never seen anything like it. We get similar pictures from the other cell lines. So these are all the cell lines. I've left out the primary, the 449 cell line from this picture, and these are our patient tumours. So very similar level of complexity. Chromosome 12 is always involved with lots of interchromosomal as well as intrachromosomal fusions. This gives us, for the first time, a really accurate picture of the large-scale structure of neochromosomes. So just focusing on 778, it contains a centrally amplified region of about 221 megabases that's highly amplified. And it's made from just 31 megabases of the genome, but harbours 289 genes. Not all of these genes are intact, many are rearranged. It's capped by low copy material from chromosomes 7 and 22. Now there are two isoforms. One has 7 on each end and one has 7 and 22. And remember, those were the things that provide us the calibration for the copy number analysis. We see similar things in the other cell lines. When we compare all the cell lines together, only 1.4 megabases of genomic sequence is recurrently amplified. And that contains 24 genes that's remarkably rich in oncogenes. So MDM2, CDK4 and HMGA2 are all there recurrently. A recently described oncogene called Yates4 is there. And we found, on the basis of the amplification and integrating our RNA-seq data, a possible novel oncogene, which had I won't focus on it, but it had a dramatic effect when this was knocked down. So a possible new oncogene emerges from this work. What would be really nice to know is how did these things form? So the sequencing results provide a clue as to what type of breakage-fusion-bridge occurred. Linear breakage-fusion-bridge would be expected to generate an excess of inversions because it's always operating from one end of a chromosome because only one telomere is lost. We don't see an excess of inversions in our breakpoint data. You'd also expect it to generate inverted duplications, and we don't see any of those. Additionally, in 778 and the 449 lines, we had the observation that in the patient primary there were rings. So this really supports the idea that a circular breakage fusion bridge is the key thing in the formation of the neochromosomes. But it doesn't rule out the possibility that linear breakage fusion bridge plays a role, and certainly in the background and on the telomeres we do see from time to time characteristic step copy number profiles that are suggestive of linear breakage fusion bridge. So that tells us a little bit about what flavour of breakage fusion bridge may have been involved, but not how this thing came together. And a simple observation put us onto a possible explanation of how the neochromosome assembled. So this shows 3 amplified regions from the 778 neochromosome. So this is the copy number profile, these are the predicted breakpoints, and I've labelled these segments A, B and C. And when we looked at these edge fusions, so the fusions that sit at the edge of these amplified regions, we noticed something quite striking. On chromosome 12, the edges were predominantly connected to other edges. So from the edge of, starting from one edge of chromosome B, if we walked across it, we would jump to chromosome A. If we walked across this, we jump to, amplified region C. If we walked across this, we jump off. And we can use this information to create a kind of assembly of something that's likely the primordial, part of the primordial neochromosome. The other thing that we noticed is that when we looked at these fusions that are not edge-to-edge fusions, they were much more likely to be interchromosomal. So the partners of these internal breakpoints were most often other chromosomes, whereas the edge breakpoints were most often intrachromosomal. This was pretty striking. When we walked out across the whole chromosome, we were able to assemble a very long stretch of it. So this is a graphical depiction of that walk, and there's over 20 amplified regions we were able to string together in this, and then subsequently there are other smaller regions that were not able to connect to this long walk. And we saw clear walks like this in 449, which is unsurprising, but also the T1000 cell line and one of the patient tumours. And while we didn't construct walks like this in the other cell lines, there was evidence that suggests that those walks existed. So these walks probably, it's the best explanation that we have, assemble fragments of the original neochromosome. It's formally possible that they could emerge as a result of breakage fusion bridge operating in a sequential manner, but that seems unlikely, and it's something that we would like to test, and I'll talk about that in a minute. Also, the dominance of these chromosome 12 to 12 fusions suggested that initially the neochromosome formed with only chromosome 12 present, and that must have happened prior to any amplification. So this kind of pattern has been described before, but never in such an amplified chromosome. It's called, and so this is a feature of some tumours called Chromothripsis, and it's another chromosome scale mutation. It's found in about 2% of tumours. The mechanism is not really known. There's a number of proposed mechanisms to explain it, but it's usually characterised by oscillations between a few copy number states, clustering of breakpoints, random rearrangement of genomic segments. And this cartoon over here is from the original Chromothripsis paper, and it shows an explanation for these patterns in the data. So we start with a normal chromosome that shatters into tens or hundreds of pieces. And then these are stitched back together by the normal chromosome repair mechanisms into something that's highly rearranged. So this is a catastrophic rearrangement of chromosomal material in a single event. Now, so our reconstruction of the early neochromosome from these walks across the the chromosomes are suggestive of Chromothripsis, and this led us to go back to the data and look for other signs of Chromothripsis. And this is something that we initially missed because we were being quite stringent about how we were calling fusions. But when we adjusted our parameters to be more sensitive and looked at the telomeric regions, which were at low copy, we found a really classic signature of Chromothripsis. And these lovely plots here are generated by Leon Di Stefano, who's a research assistant in my lab. And we've taken to calling these Campbellgrams after Peter Campbell. These plots like this appear in all of their papers. So this shows the copy number profiles for the telomeric caps of the 778 neochromosome, and then the rearrangements that are present, which are really classic signatures of Chromothripsis. So we have only a few copy number states. Essentially, chromosome 22 just has a single copy number state. Chromosome 7 has some deletions, and it has 2 copy number states, really, or 3, including the deletions. These regions present at 2 copies, and then single copy regions. And importantly, you see these rearrangements at the top. They're split into different classes of rearrangement. The D stands for deletion type, the TD tandem duplication, and then these are 2 different kinds of inversions, essentially just pointing in different directions. And these are in equal proportion, which is one of these statistical characteristics of Chromothripsis that needs to be satisfied. And then looking at other chromosomes, these are chromosomes not involved in the neochromosome but sit in the background, we see other signatures of Chromothripsis. But although you can't be sure here because we haven't looked at fusions, we don't have that power, but you see oscillating copy number states between just a few states. And this is evident on multiple chromosomes throughout these tumours. So, what would be really nice is if we could test this in some way. So how can you test whether Chromothripsis occurred prior to breakage-fusion-bridge? We're not observing the event itself, and unlike the original analysis of Chromothripsis, which was done in what was essentially a very quiescent tumour genome, there's no copy number amplification in those original observations of Chromothripsis. We've got all of this amplification superimposed on top of what we think is a chromothriptic event. So how do you explore that? And can the patterns that we observe be explained without resorting to saying that it's all kicked off by Chromothripsis? So to do that, a postdoc in my lab, Vincent Corbin, developed a computational model of breakage-fusion-bridge to explore its dynamics. So this is sort of a snapshot of the cartoon that I showed earlier of how breakage-fusion-bridge works, and it's really at the heart of this model. So we have a simulation that can do this, that can start with a dicentric ring, randomly simulate a pair of DNA breaks, generate 2 daughter rings, and then track them. So the key bits of the model are really that these DNA breaks here are generated from a uniform probability distribution. And without doing anything else, if we just run this model, this process chews away the neochromosome, or these rings, and you end up with nothing. It just completely erodes them away. So we need to add something. We need to add selection to the model in order to retain some material. And it's natural to think that selection might involve amplified oncogenes. So what we do is assume the fitness is proportional to the number of copies of some key genes, in this case oncogenes, and we can select those genes in different ways. Ultimately what we did was we used the recurrently amplified material in the genome as the selected regions. But we tried other things as well. We require that the model conserves at least 1 copy of these selected genes. And then we calculate a fitness, essentially by counting how many copies of each of these genes is, but with some saturation, so not allowing fitness to become infinite. Well, then what we do is we just choose 1 daughter cell randomly, weighted according to the fitness. And in doing this, we're kind of exploring the possible dynamics of one lineage through a tree of possible fates for the neochromosome. This shows a very quick cartoon of what happens. So we started with chromosome 12, just having lost both telomeres, and we're at one copy. And then we have, in this case, what we've actually done is there's a Chromothripsis first, and then we start amplifying. And so very quickly, so this is after 100 cycles, we've got quite a high level of focal amplification. And by 500, we have something that kind of looks, at least qualitatively, like the patterns we're seeing in our data. But that's not really enough. It's followed only one daughter at every cycle. So what we do is we run this model thousands of times. In each run, what we do is we follow one daughter, as I explained before, and this selects essentially one possible lineage and ignores the others. We run it thousands of times and we generate summary statistics from each run. For example, the number of genomic intervals present on the neochromosome in the model, the genomic interval length, and we also explored other summary statistics of the run. So this is called an ensemble model. We all played around with different fitness and selection models and different distributions for breaking the rings, and we ran the model with and without Chromothripsis. The nice thing, the model evolved eventually to the point where we used empirical data to model Chromothripsis, and this is the first time this has been done. Previously, there's been a small amount of modelling of Chromothripsis from the Sanger Institute, and they've always assumed uniform distributions, while stating that breakpoints due to Chromothripsis should be clustered. So we've used, a nice feature of this is that we've used empirical data to refine the model. So running the model with breakage-fusion-bridge only gives us, generates plots like this. So this shows the number of genomic intervals over what's essentially time for 1,000 runs of the model. And also plotted on this graph are the number of genomic intervals for each of our cell lines. The first thing we did was, with breakage-fusion-bridge only, tried out many different models of selection and distributions of breakage to try and fit this model to the data, and we simply couldn't. Then we added Chromothripsis, and immediately we were able to fit the model. So fit the model to the data. So this was very encouraging. And what emerges from both looking at in detail at the copy number analysis and also the fusion data that we generated from the neochromosome sequencing, and then also the modelling results, and as well as low resolution studies, we're able to sort of synthesize a life history for these neochromosomes. So this cartoon shows how we think they evolved, what the evidence from all of these studies suggests. So there's strong evidence that Chromothripsis of chromosome 12 leads to the formation of a ring chromosome. And then at that point, the neochromosome enters an amplification and deletion phase in which breakage-fusion-bridge amplifies oncogenes while deleting away extraneous material. The patterns of fusion suggest that new material from other chromosomes is added in a punctuated way, so not all at once, but one chromosome at a time. And then the other thing that I've alluded to but not shown any data from is the fish and chip studies showed that what can happen in many of these cell lines is that the centromeres are chewed away by the process of breakage-fusion-bridge. and that this leads to a crisis in the ring that needs to be resolved in some way. There's two ways of resolving it, and we observe both outcomes. One is you capture a native centromere from another chromosome, and the other is you form a neocentromere. And we observe both of these things. And then finally, the neochromosomes are stabilised by capturing these telomeric regions from other chromosomes. And it's quite likely, well, certainly these telomeric regions, we've observed that they're chromothriptically derived. It seems likely, although we don't have direct evidence for this, that this acquisition of new material it also involves chromothripsis. So these cells really put up with a staggering level of genomic instability. It's really remarkable. So this has provided insight into the formation of neochromosomes, but I think the approaches that we've used are likely to have broader relevance. And in particular, it's notable that the emergence of resistance to targeted therapies is sometimes, but not always, driven by extreme amplification of the target. So this happens in melanomas that are treated with BRAF inhibitors. Mark van Delft's been working on ABT737 resistant fibroblasts, and he gave us an array CGH copy number profile from one of these, from several of these fibroblasts, and this very striking pattern emerges. And so this is ABT737 targets BCL-XL, and it's highly amplified to between 20 and 60 copies. And this pattern is really evocative of Chromothripsis and linear breakage-fusion-bridge. Now, we can't tell here because the resolution is too low, and to definitively see this, you need sequencing data. But this is quite suggestive of a chromothriptic event followed by linear breakage-fusion-bridge. So I think there's potentially broader relevance of these methods. So briefly now, I'd like to talk about another method of predicting breakpoints that was motivated by a different data set. So the idea in this project was to study the evolution of tumours in transgenic mice. And the idea really was to sequence the tumour and then look back through serial blood samples in order to track evolution. Now, the transgene is really the very first hit. So our focus was really in trying to map where the transgene inserted precisely, what was its structure, did it go in one copy or multiple copies? So it's really to characterise this very first hit. And to ask the question, are there other consequences of the insertion? And the answer turns out to be sometimes there is and sometimes it's very clean. So we've now done a couple of transgenic mice, but I'm just going to focus on work that we did on the Eu-myc mouse tumour. So Jan Schroeder, a postdoc in my lab, did the analysis here, and this was a project in collaboration with Ricky Johnston and Richard Tothill at the Peter Mac. So they sequenced the Eu-myc lymphoma genome, the germline of an Eu-myc mouse, and also a WEHI Black 6 mouse. They used 100 nucleotide paired end reads sequenced on an Illumina HiSeq. And unfortunately, the average fragment size of these reads was only 213 nucleotides. So this little cartoon here shows what that means. This is a fragment of DNA produced by sonicating the whole genome and generating these fragments. These are amplified and then size selected. And the size selection was just a tad small, really. So this fragment size is, on average, 213 nucleotides. 100 nucleotides of each end are sequenced, leaving only a 13 nucleotide gap, on average, in the middle. And that gap's really important when you want to map fusions in a genome, at least using the kind of paired end methods that I talked about before. This means that 50% of the reads actually overlap. There's no gap at all in 50% of the reads. And so for those methods, that's an incredible loss of sensitivity. And this is, so this was a problem here, but it's a generic problem. If you deal with FFPE samples, they'll be highly fragmented and you'll hit this problem. And as read lengths grow, this is increasingly going to become a problem. So I'm kind of putting up the answer straight away. This figure looks remarkably like a figure in Jerry Adams' paper 27 years ago. So this is the structure of the Eu-myc transgene. Most of these fusions can be inferred from that paper, but not at single nucleotide precision. The new thing here is the mapping point, which is over this side. And I've labelled all of the fusions, and then we applied a bunch of different algorithms that are published by other groups to try to call these fusions. We used breakdancer first, and that worked pretty well. It was able to map most of these fusions in the transgene. But really critically, it couldn't get the ones on the ends, which are probably the most important. It seems to be a theme for breakdancer. We tried another technique called DELI, which was developed in the context of the 1,000 genome data for identifying structural variants in populations. And it was only able to find one fusion, which turns out to be, it's the least interesting one. It's a fusion that occurs because of the circular nature of the PUC12 vector. So it's not even interesting. It's just linking one end of the vector to the other, and found nothing else. And that's a direct result of this very small fragment size. So DELI uses the paired end fragments to guide where it calls breaks, and then does single nucleotide analysis at the breaks. CREST is another single nucleotide resolution method, and it did pretty well. It's not able to map what's going on at this end of the transgene, but it's the only method tested so far that actually located the transgene in the genome. So this set us thinking about how do we make sense of this data. We have a loss of paired end information, and so we decided that we would focus on these things called split reads. So before I showed a cartoon with reads spanning the breakpoint but not touching it, when the reads themselves span the breakpoints, you'll get a little bit of read from one chromosome and a little bit of read from another chromosome. Now, some next-gen sequence aligners, what they will do is they will simply mask out those bits that come from another chromosome or come from another locus. This is called a soft clip. A soft clip is like a special mismatch state that is applied just at the ends of reads. So we identify these soft clips and then realign them to work out where the other side of the fusion is. And we do that on both sides and build a consensus coming in from both sides. So this is a slightly noisy operation because we can be dealing with quite short fragments of DNA in doing this, but it seems to work quite well. And importantly, what we do is we take into account possible microhomologies and also untemplated sequence that are inserted at the fusions. And we were the first method to do that. It makes quite a big difference. Socrates is also the only method that can deal with heterogeneous or promiscuous breakpoints. And we did that to be complete, but it turns out to be quite important in the Eu-myc mouse transgene. So just to compare to other methods, at 30 times coverage, this method has the highest sensitivity and equal highest precision, and it's an order of magnitude faster than other methods. And I think we can probably speed it up further. So Socrates gets all green ticks. We're able to identify all of the fusions and in particular figure out what's going on at this end. And so there's actually a loop back to the start of the transgene, and from copy number analysis you can reconstruct how many times the transgene is present in a cassette. We're able to develop models of what the transgene cassette looks like in the germline. So this is a picture of it. So these Ts are the transgenes, and they're present in 3 copies. So we've got 5 copies of the Eu-myc gene, 3 coming from the transgene, and 2 normal copies. And then this is connected to a region of chromosome 19. It's a region that interestingly contains the JAK gene. And unfortunately, this copy number region kind of runs out at a repeat, and so we're not completely able to map it, even with mate pair information. There's frequently amplification of the transgene in Eu-myc tumours, and so this is a model, and we're not completely sure that this is put together correctly, but this seems to happen with reasonable frequency in Eu-myc tumours. So, with a more sensitive approach, we're able to map the Eu-myc transgene. We're applying this in other cell lines, other transgenic mice. This sort of weird data that we got from the Eu-myc mouse motivated us to develop a new method that's generically usable. It actually performs generally very well. And it's probably its greatest asset is its sensitivity. So you can adjust the parameters of the method so that it's extraordinarily sensitive, and then used in conjunction with other kinds of information like RNA-seq data or copy number or prior knowledge, as we had with the Eu-myc data, can very sensitively find fusions. And this is being extended by Daniel Cameron, who's a PhD student in my lab, to incorporate the other kinds of evidence that are also available that we just ignored in this case. So I've described some boutique methods and some generic methods that we've developed to help us make sense of tumour sequencing data. I've talked about these interesting neochromosomes, and we're able to infer the life history of them from sequencing data and also from mathematical modelling. But these approaches that I've talked about, they're generically applicable. We're using them now more and more in tumour genome data sets, but now plasmodium sequencing, for example, is really taking off at WEHI. And exactly the same approaches of copy number analysis and rearrangement detection are applicable. It just remains to thank collaborators on these projects. So the Liposarcoma project was really a very close collaboration with David Thomas at the Peter Mac over quite a long period of time, and involved multiple members in my lab, so including Vincent Corbin, Arthur Sue, who's a postdoc who's now left, Leon De Stefano and Jan Schroeder, as well as others. The Socrates work was also a collaboration with a large number of people. From the Peter Mac, Ricky Johnson and Richard Tothill provided Eu-myc data, Mark Shackleton and Sam Boyle provided melanoma data, and Jan, Arthur and Daniel did much of the work. Also like to acknowledge Jeff McIntyre and Marek Tomero from NICTA, or formerly from NICTA. They were very enthusiastic about Socrates and applied it in prostate cancer. And then I'd like to thank all of the members of my lab, as well as the other lab heads in the Bioinformatics division, Terry, Melanie and Gordon, and Matt Wakefield, who is, I guess, a part-time member of our division, for very helpful conversations in this work. So I'll be very happy to take questions now. Thank you.
So why if we return to the first part of your talk, why do these giant chromosomes occur in sarcomas predominantly? Why chromosome 12 and the other two linked?
So chromosome 12, because it happens that MDM2 is there essentially. MDM2 is almost always amplified in well-differentiated liposarcomas. So that's the reason. It's essentially selection.
It could be other oncogenes.
Sure, it has to be an oncogene that needs to be amplified, and certainly other oncogenes do like to be amplified. It's very clear that it's this set in liposarcomas. Perhaps in other kinds of tumours, it's other genes. The real question is, what is the underlying genetic defect that allows these things to start? So while the neochromosome may drive the tumour formation, something underlies its formation, and that we don't know yet.
I think you kind of answered my question there, but it's a chicken or the egg question, really, isn't it? Like, what is it the genomic instability is the first lesion that occurs, and then that is just a result of it, or is it the catastrophic shattering that is the start point for the tumour?
Yeah. So
you can't really tease that out, I assume, unless you have.
Yeah. So it'd certainly be nice to undertake a larger study that has the power to look at germline defects and other defects in the tumour genomes to try and tease out what might be the real initiating event. This study was never really designed to do that.
We have 23 pairs of chromosomes, and I know there's an Australian bull ant that only has one pair of chromosomes, so the male only has one chromosome. So the question is, why do we have a certain number? Is there an optimal number? Is it a random number? What's the best number?
That's a terrific question. I don't have the answer for it. Obviously, marsupials have beautiful chromosomes, and I've spent a long time working on those. They have very few chromosomes, and perhaps that's nicer to work on. I guess the interesting thing here is the way, that frequently tumours have more than 46, that this aneuploidy is perhaps a first step in the process of becoming genomically unstable and forming new chromosomes.
For the second half of the talk, we kind of assume that transgenes are genetically stable as we breed the mice with the Eu-myc, but we know there are some weird things. So if you inherit an Eu-myc transgene from the mother rather than from the father, the animals, the offspring get the tumours faster. So have they in the study looked at the germline following different transmission? And so maybe the answer is that it gets amplified following female transmission, something genetically different happens.
Yeah, the study wasn't. So I never even found out what the sex of these mice were, although it's something that we could easily figure out. The study only sequenced one Eu-myc germline and tumour deeply. So more deep data would be nice. We did a bit of low coverage sequencing, but it's of much less value. So that doesn't emerge from the data that we've got. It'd be terrific to get more data to try to tease those things out.